Welcome to all of you. Good morning, good afternoon, good evening, wherever you are. Today we have two speakers, Dr. Daniel Huster and Dr. Olga Gursky. The first speaker is Dr. Daniel Huster. He received his bachelor's degree and diploma degree and PhD degrees from, and also initial postdoctoral training from Leipzig University in Germany. Part of his, actually all his thesis work was carried out at NIH under the guidance of Dr. Klaus Goerich. Then after completing 2 years postdoctoral research at Iowa State University in Dr. Mei Hong's laboratory, he went back to Leipzig University to start his independent research group in 2006. Since 2008, he has been the head of the Institute for Medical Physics and Biophysics at Leipzig. His research has been focusing on the structural biophysics of membrane proteins and amyloid aggregation, and also on the development of NMR spectroscopy as well. So with that brief introduction, I would like to welcome Daniel to get started. Thank you very much for joining us, Daniel. Go ahead and get started.
Yeah, thank you very much, Rams, and hello to everybody out there. Yeah, it's great, Rams, that you organized this very exciting series of talks and discussion meetings in those difficult times still. And we still hope that we'll have a chance to see each other in person soon and go back to the normal discussions at posters and so forth. Yeah, thank you very much for the invitation to speak here. So today I'm going to focus a little bit on our efforts over the last 10 years on the structure and dynamics of mutated amyloid beta fibrils. And about 10 years ago or so, when we started with that project, the question that really fascinated us was, since there is such a large variety of soluble proteins in all kinds of different folds and structures, one can almost find conditions under which these well-folded proteins go into amyloid fibrils. And then the large structural polymorphism is basically abolished, and we always end up with a cross-beta structure. And this transition seems to be possible for almost all proteins. And this led us to ask the question, is there perhaps somewhat of an inherent property of many proteins, if not all, to undergo this structural transition from the well-folded soluble three-dimensional structures into the insoluble beta fibrils? And so we asked, what is the influence of local perturbations or local interactions, physical fields or so, on the fibrillation process and pathway? And at the time, the A-beta 40 structure was, it wasn't really a structure, it was more like a model. Shown as this hairpin. Of course, now we all know that there's also extended models, as we heard from Rob Tico about 2 or 3 weeks ago in this meeting here. But this was basically the structural model we had at the time. And we looked at it, and we looked at the stabilization, intermolecular contacts. And there were quite a few that were described. And the one that was described in all the studies at the time was the intermolecular contact between phenylalanine 19 and leucine 34. And so we said, well, let's just try and modify that hydrophobic contact here and observe what's going to happen to our three-dimensional structure of the A-beta protein and report structural dynamical changes, do the known assays to study morphology of fibrils and so forth. So in other words, we introduced local perturbations in the structure and wanted to observe how fibrillation kinetics, structure, and dynamics is influenced. So this was the first set of mutants that we generated. And in the native A-beta structure, we have this contact, which is a phenylalanine and the leucine. So this is a hydrophobic contact. And then we said, let's just modify it. Let's just challenge the structure. And the first idea we had was we introduce a glycine side chain, which introduces a lot of flexibility. So if you look at the Ramachandran diagram, of course, we have a lot of structural freedom, or we introduce a mutant that abolishes this flexibility by introducing this proline here, which has minimal flexibility. Only the cis and trans would be possible. And then we said, well, instead of the hydrophobic benzene ring, we could also introduce a charge here, a negative or a positive charge. We also made a mutant where this benzene ring had this hydroxyl group here to allow for hydrogen bonding. And then we said, maybe we can even replace the hydrophobic contact by electrostatic attraction in a salt bridge, by having a lysine residue in position 19 and a glutamate in position 34, or see if we have electrostatic repulsion, whether or not this abolishes the property of the A-beta peptide to form fibrils. And now you can place your bets and see which of those modifications still form fibrils. The answer is all of them did, which was a little bit disappointing at the time. So you see the electron micrographs show that all these mutations showed very nice fibrils, except for the F19 K19 mutant. So this is basically the salt bridge. So we learned at that point that the hydrophobic contact cannot be replaced by a salt bridge. Well, meanwhile, we also found conditions where this mutant here forms also very nice fibrils, but at the time we didn't have this knowledge. So all of these, as I said, form fibrils. They vary a little bit in their morphology, but by and large, they were just nice, pretty fibrils. And then, of course, we studied the THT fluorescence to observe the growth curve and the kinetics of fibril formation by using the thioflavin T fluorophore, as anybody else does. And well, by and large, in our hand, the wild type fibrillation time is about 10 hours, and most of our mutants had a similar fibrillation time. There was the proline mutant that took about 10 times longer. The glutamate was also about 5 times longer, but by and large, the fibrillation ended up in the same morphology. Also, the X-ray diffraction, this is not the most pretty X-ray diffraction pattern that we gathered at that time. We can do this much better by now, but also this basically gave the result that all our fibrils from the mutated peptides form very nice fibrils of a very similar morphology and structure. And then we also looked into secondary structures. So we labeled 4 residues in our peptide sequence. So this is valine 18, which was carbon 13 labeled. This is phenylalanine 20. So this was one before the mutation. This is one after the mutation. Then alanine 21, 2 after the mutation, and glycine 34. And we did solid state NMR spectra, as also reported by many, many people, Rob Tycho and many, many others. And of course, the chemical shifts that we observe are indicative of secondary structure. So if a random coil gives you certain backbone chemical shifts, then certain secondary shifts, C alpha and C beta moving to the left and C beta moving to the right, indicates that we have a helical structure, and the opposite movement indicates that we have beta sheet structure. So this is the result. It's going to be a little bit busy. Let me just go through it step by step. So this is the chemical shift of valine 18, phenylalanine 20, alanine 21, glycine 33. And those are the canonical chemical shifts that you observe for alpha helical structure, random coil structure, and beta street structure. So this is the wild type A beta. You see all these residues, valine, phenylalanine, and alanine, show very nice beta strand structure, as you would expect. The glycine is a little bit inconclusive because the secondary chemical shifts are not so easy to pick up. So we focus on those here. And now we can go through all the mutations that we looked at, which are listed here, and see how each residue responds to them. So we first look at the valine 18. So this is the residue just before the mutation. And here we have the mutation, the phenylalanine 19 to glycine, proline, glutamate, lysine, tyrosine. And you see valine 18 doesn't care. So no matter what you do at phenylalanine 19, valine 18 remains very nice and beta sheet structure, right? So now we look at the phenylalanine 20, the residue after the mutation. So this would be this row here. Yeah. So if we put in a tyrosine instead of the phenylalanine, then we still see better sheet structure. If you have a proline, glutamate, or glycine, then you see our structure becomes more loosely defined. So it's not beta sheet, it's more random coil. And if you put in the lysine, then we see a large chemical shift change of 8 ppm or something like that, all the way to a local alpha helix structure. Yeah, it's just a local structure in the backbone. Of course, it doesn't mean that we have an alpha helix, but it's just a local torsion angle set that is compatible with alpha helix structure. So then we move one forward. This would be alanine 21. So if we put in a glycine, a glutamate, a tyrosine, or a lysine in position 19, you see basically nothing happens. So the effect of our mutation is totally forgotten 2 residues down the sequence. Only the proline, which we put in position 19, somewhat alters the chemical shift of alanine. And then finally, we have done the mutations of the glycine 33 of the phenylalanine and observed the glycine 33 in the opposing strand, and by and large there is no structural polymorphism anymore. So by and large, the result of this analysis is that no matter what perturbation we put in, the canonical cross beta structure of A beta 40 is very robust against all mutations that we've introduced. So for all these different mutations, we could find fibrils, except for the one where the hydrophobic contact is replaced by the salt bridge. But as I said, meanwhile, we also found conditions that this mutant also forms very nice fibrils. So that means that local structure dynamics, fibrillation kinetics is very similar, or in other words, the A-beta structure is very robust against whatever modification we do. That was a little bit disappointing for us. However, we hadn't really looked at biological effects. And I should say that the position 19 and position 34 are positions that we chose more or less for physical arguments. So there are no known early onset A-beta amyloid diseases or anything related to these positions. Yeah, and they were just purely chosen out of physical reasons. However, we did biological experiments with Sudipta Maiti. This was during my first sabbatical in Mumbai in 2014. And Sudipta is a brilliant biophysicist who also does a lot of cell studies. And he suggested we should look at those modified A-beta fibrils and see if they're toxic or not. And this is the result. So we checked the cell viability here in primary cortical neurons, red neurons, yeah. And so we add a certain amount of A-beta. So this is the control, this is the wild type. And after 60 hours, we read out how many of the cells survived. And with normal A-beta 40, you see that about 40% of the cells survive. However, when we checked all our mutations, you see that the toxicity is basically abolished. And there was essentially no toxicity left for these mutants. That was very interesting because those were mutants that were not reported in any disease or any physical condition or so. Well, Sudipta went ahead and also asked for the reason why that could be. And since all our structures were so similar to the wild type structures, he was suggesting that it's perhaps the oligomers and the oligomer interaction with the cell membrane that could make a difference here. And sure enough, he measured the binding of A-beta and all our mutated A-beta variants to liposomes. This is phosphatidylcholine, phosphatidylglycerol, and cholesterol 1:1:1. And he saw that while A-beta wild type binds to these liposomes, all the mutants did not essentially. And that could be another hint that it's essentially the oligomeric structure that is responsible for these tremendous differences in the toxicity of those elements. So I'm more a physical chemist or a biophysicist. So for me, it was further interesting to see what structural consequences these mutations would have. And this again is our leucine 34 F19 context. And we wanted to study the one mutation that gave the largest deviation from the canonical secondary structure that we had observed before. And this is the F19 K19 mutation. So we replaced the phenylalanine by lysine. And of course, the lysine is a positively charged residue. So if you look at the difference in free energy, then very simple hydrophobicity scales will tell you that you have about 4 kcals per mole of unfavorable hydrophobic energy when you introduce this charged side chain here. And the first question, of course, was, is this really a charged side chain? Because it could also be deprotonated, and then we would perhaps not have to pay such a high energy penalty. But we could measure that also very well by NMR. So this is the chemical shift of the side chain of K19. And there are reference values for a protonated side chain and a deprotonated side chain. And close enough, or sure enough, we see a protonated side chain. So this side chain must be charged. Well, then, of course, you would still wonder if that could be somehow accommodated in this hydrophobic interior of the fibril. And we were thinking, well, this could also only work when the side chain is sort of like moving out of the hydrophobic center. So one could, for instance, imagine that the two somehow change position, which of course would have consequences on the secondary structure here. So this would introduce tremendous local perturbations of the structure, and put this F20 in close vicinity to leucine 34, and the K19 would then phase outwards. This would be one scenario. Or if you look at the hydrophobic and the distribution of hydrophobic and hydrophilic residues, you could also imagine maybe the entire beta strand is swapped, because this conformation, if again I go by a hydrophobicity scale, is more favorable than this one here. Yeah, so those were two scenarios that we thought we could take a look. So how could we study that? Well, of course, we could now check if there was a contact between phenylalanine 20 and leucine 34, for instance. So NMR gives us the possibility to check for these interresidue contacts. So if the local secondary structure is changed such that K19 and F20 are swapped, then the F20 would be in close proximity to leucine 34, and we should observe a contact. Also in this situation where we have the entire strand being flipped, the L34 should be in close proximity to F20. So this is something we could check. The other decisive feature or structural feature is this salt bridge here that we have between aspartate 23 and leucine 28. As you see here in our left model, the K28 would still be in close proximity to the aspartate 23. So we could see if that contact is still there. In the situation over here, we would not have this salt bridge to the lysine, because if you flip the entire strand, then the lysine, the aspartate would face the exterior of the fibril. However, luckily, this glutamate then would be in hydrogen or in salt bridging distance to the K28, and possibly if that was the correct structure, we could observe a salt bridge between K28 and glutamate 22. Okay, so here's the results. First question: in the F19 K mutant, is there a phenylalanine 20 leucine 34 contact? And do the carbon-carbon correlation experiment, and we can sure enough pick up this contact. So this actually is the scenario that we find. So now all of a sudden the F20, which normally faces the side chain to the exterior, now is switched to the interior and can make a contact with the leucine. So that we find already. Then we do all this secondary structure analysis. I have just color coded it for time reasons so you can look at it much easier. So in orange, you see better sheet structure. This is the wild type. And if you look at our mutant, then you see that the colors are much different. So we see random coil structure, and especially here around F19, where we now set this K19, this leucine mutation, and we see a lot of structural perturbation. Significant secondary structure perturbation. And finally, the question was, do we see a salt bridge between K28 and either D23 or E22 in the two models? And we do for this, we do redo experiments between the side chains of the glutamate or aspartate, the CO group, and the NH3 plus group of the lysine side chain. And in the wild type, we see this nice redo signal here, which tells us that there is a salt bridge. But in our mutants, we neither see a salt bridge with D23 nor with E22. So we also did some MD simulations with Felix Hoffmann from Halle, where we could essentially recover all the experimental features. So the experimental findings were that we do see that K19 is positively charged and pointing outwards towards the aqueous phase. We see that in the simulations. We also see the F20L34 contact as we saw experimentally. We see the significantly perturbed lower beta strand, which we see in the left model, not in the right model, where we just swap However, the MD model still predicts the salt bridges, which experimentally we did not find. So since we said rather minor and conservative mutations, but they turned out to have tremendous biological effects, essentially abolishing the toxicity of all our A-beta species, we're wondering what would be the minimally tolerated perturbation of the F19L34 contact. And to this end, we designed 3 new mutations, and those are mutations on leucine 34 now. So instead of the leucine, we introduced the D-leucine stereoisomer, an isoleucine or a valine. And I'm not going to go through all these data. Here's the paper. You can read if you're interested. But the bottom line is there were minimal perturbations of the A-beta structure, and the toxicity was close to wild type. So these minimal modifications of leucine 34 basically did not introduce much alteration of the toxicity. Then we turned our attention towards the phenylalanine 19 again, and we did a series of modifications of phenylalanine benzene ring. So those are non-proteinaceous side chains. This is the cyclohexane moiety. This was first introduced by Katsumi Matsuzaki from Japan. then we have here a phenylalanine side chain that is 1 carbon shorter than the phenylalanine. Here we have a side chain that is 1 carbon longer. And we also, instead of the benzene ring, we introduced a naphthalene moiety. And again, I spare you all the data, minimal perturbations in a better structure, but significant decrease in toxicity. And this put all together, so here you can read it if you want. This introduces the idea that the phenylalanine 19 is the most crucial site for toxicity, at least in the set of mutations that we've looked at. So the most recent work that we've done is to again challenge this contact between the phenylalanine side chain and the leucine side chain by shifting the register in the backbone. So move the phenylalanine to the right and the leucine to the left. And we did that by introducing, again, non-proteinaceous amino acid residues. So this is the phenylalanine wild type, where you have the N, the C alpha, and the CO. And here we introduce backbone modified amino acid. You have the N, the C alpha, then you have an additional carbon which extends the backbone by 1 carbon, and then here's the CO. And we also have a residue, or we also have an amino acid which has now 2 extra carbons. So this would be the phenylalanine side. We have the same amino acid building blocks also for the leucine side. So we have a chance to put the fibrils out of register by 1, 2, 3, 4 extra bonds. And this is our most recent paper on that subject, scientific report from last year. Again, we see very minimal perturbations of a better structure. So somehow this structure can adapt to all these modifications, and also no major differences in toxicity. And the last one, we probed the intermolecular hydrogen bonding at the phenylalanine 19 leucine 34 site by abolishing the ability of the residues to form these intermolecular hydrogen bonds. And for this, we followed an idea that was first put up by Stephen Meredith, who also talked in the symposium here. And we used, so they used different amino acid residues. We used, again, the phenylalanine 19 and the leucine 34. And we used these building blocks that basically had a methyl group here at the amide, which abolishes the possibility for the intermolecular hydrogen bonding. So we found that one methylation site, so either the phenylalanine or the leucine, significantly slowed down fibrillation. And if we have both building blocks exchanged, then the ability to form fibrils was completely abolished. And this is also in this manuscript by Benedikt Schwarze. So this is a summary of all the mutations. Of course, we've done a lot more mutations than I talked about in these 20 minutes so far. But essentially, we were focusing on the phenylalanine 19 and the leucine 34 contact and doing all kinds of modifications. We also focused our attention to the N-terminus, where we have pyroglutamate modifications and similar things. The summary and the bottom line of all of what I talked about is that the A-beta fibril structure is very robust against whatever mutations we set. But we do see some structural changes, local structural changes on the secondary structure level, a little bit on the morphology, but overall the structure is very robust against any of the mutations that we introduced. However, the F1934 contact is crucial for the toxicity of A-beta fibrils. And so far, we would suggest that the most susceptible one is the phenylalanine 19 site. Of course, again, none of these is a reported mutation that causes an early onset Alzheimer's disease or anything like that. But we looked, of course, in the genomic data that's available, and there is indeed a phenylalanine 19 leucine SNP reported, so it has a given number. But we have no idea if there is a physiological important, or if that person who had this mutation developed early onset Alzheimer's or anything. But nevertheless, we would suggest that whatever small molecule binds to this F19L34 contact could be considered pharmacologically relevant. Overall, the local fields alter the energy landscape of fibrillation, but the general cross-beta structure is retained in all these mutants, and that was quite interesting. Now, in the last 5 minutes or so, I'd like to just briefly talk about some completely unrelated subject, and that concerns the growth curve that we see normally in an ensemble where we have a superposition of monomers, small oligomers, maybe some protofibrils, some fibrils, and everything like that. So this is rather heterogeneous, and the growth of the structures is very insynchronized. And if you want to study individual fibrils and want to see how they grow, there's not so many options that we have. Of course, we can observe those things in a confocal volume, but the species will diffuse through the confocal volume faster or slower, and we only have a limited amount of time to observe, for instance, the growth of these structures within the confocal volume. And we teamed up with a brilliant physicist here at the University of Leipzig. This is Frank Zichos, who was building a so-called thermophoretic trap, and he trapped all kinds of structures, small beads or polymers and things like that. And then also the idea came up maybe to put amyloid fibrils into these traps. So what are these traps? So here's just a cover slips with a chrome ring and a certain diameter of, I don't know, 6 microns or something like that. And the force that traps an A-beta fibril into this chrome structure here is the so-called thermophoresis. So if you have a temperature gradient between a hot and a cold environment, and a small particle picks up this drift velocity, which is proportional to the temperature gradient, and then there's a so-called Ray coefficient that describes all that. Now this is the geometry. You have two cover slips, and here you have this chrome ring, and we want to trap an amyloid fibril within that chrome ring structure. So they diffuse, of course, through the aqueous medium. When we have it in the structure, then we switch on this infrared laser, and this infrared laser now heats the chrome structure, and it rotates around the structure with a frequency of 100 Hertz. They have also built lots of setups with a feedback mechanism and anything like that. But just the simple setup of rotating the laser clockwise or anti-clockwise on the chrome ring is sufficient to trap the amyloid fibril within the structure. And so this is our trap here. So you have a temperature increase by, I don't know, 20, 25 Kelvin or something. And this is the temperature profile, which you can very well describe. It's almost harmonic. And the fibril then is trapped within this volume of the trap. And here you see the probability of the trap. And here I show you some images. This is a fluorescence image of a fibril, A-beta fibril. And the staining is just thioflavine T. So it's very simple. I mean, the thioflavine that you use anyways also gives nice results here for as the fluor for the staining. And this is very robust. I mean, here we observe over 10 minutes and you see there's no bleaching, nothing. So you can observe this even over hours. And then you can go ahead and do a stepwise analysis, and you can observe the motion of the fibril parallel and perpendicular, and you can study the translational diffusion. You can describe this very well. Also the rotational diffusion you can describe very well. And then you can look at the relation of the translational diffusion coefficient and the rotational diffusion coefficient with the length of the fibril. And it turns out that the translational diffusion coefficient is inverse proportional to the length of the fibril, and the rotational diffusion coefficient is inverse proportional to the third power of the length of the fibril. And that is of course great because it gives you a chance to observe length changes of the fibril within the time when the fibril is trapped in the thermophoretic trap. Yeah, and so this allowed us, in the presence of peptide monomers, to observe the growth of seed fibrils in the presence of the peptide monomers. So this is a certain fibril that we trap in the thermophoretic trap here, so at a certain salt condition, pH, and so forth. And you see the rotational diffusion coefficient decreases, and we can, from this decrease, calculate a growth rate. So this amounts to about 0.06 nanometer per second, or one monomer is attached every 16 seconds. We also saw some fibrils that did not extend, yeah, under the same conditions. And if we increase the salt conditions, decrease electrostatic repulsion, we could also see a faster growth rate. So here we have a monomer attached every 6 seconds or so. And this all, of course, fits very nicely with the cubic proportionality of the rotational diffusion coefficient with the length of the fibril. We've also done other conditions. Here you see different pHs. We've also done more biologically relevant fibrils, pyroglutamate A-beta. Here we have A-beta 42. So this can all be very nicely described in the trap. And finally, we can also start observing secondary nucleation or things like that. And then you see that here. So now you've seen that, maybe you've seen that a little part of the fibril is released and diffuses out of the, here, this was just the event. Yeah, so this comes back in over and over and over. Yeah, and this is basically shown after about 120 minutes, after 2 hours. This is when how we observe the length of the fibril. There's no monomers around, that's why it remains the same. But then here we see this event of fracture. So this is now again in the steady state. So here you see the breaking of the fibril, the little fragment that goes off. This is of course too small to be trapped, so it diffuses out of the fibril, and then we observe the shorter fibril, which of course has a higher diffusional rotational diffusion coefficient, and we can start observing these secondary effects, maybe also secondary nucleation, things like that. So this all was published about 2 years ago. And with that, I think I'm at the end and my time is also up. So thank you very much to Juliane, Alexander, Benedict, and Holger from my group for all the work. And then I mentioned the great collaboration I have with my Indian friends, Madhu and Sudipta at TIFR Mumbai and Hyderabad, Felix Hoffmann acknowledged for the MD simulations, and then Frank Zichos is the person, the smart physicist who is doing all this great work on the thermophoretic trap. And with that, I'm at the end. I'd like to thank Rams very much for inviting me. Thank you all very much for listening to me.
Thank you very much, Daniel, for a great talk. Please post your questions in the Q&A folder of Zoom. It will be read and discussed. Daniel, let me get started here with the question. You know, phenylalanine Phi interaction seems to play a big role based on Woody Gazette studies on A beta aggregation. So that could be a nucleating factor to form toxic oligomers. So this is just a hypothesis. Somebody might have tested it. So what's your take on that?
Yeah, I mean, absolutely. I think small molecules with aromatic structures also could really interfere at that position. They could do some pi stacking with the phenylalanine, and in all our modifications, the phenylalanine 19 was most prone to was the residue that was that reacted most to all the modifications, and the toxicity was really impaired as soon as we touched the phenylalanine 19. So you have a small molecule that can preferentially interact with this site. Could also be a cation pi interaction that could play a role here. I think this could be definitely of pharmacological relevance. So I think this is a very valid point, of course. Yeah.
So am I correct that all of your toxicity measurements were on fibrils? Did you check on monomers and oligomers?
Yeah, well, I mean, we just add monomer solutions to either cells or the primary neurons, and we weigh a sufficient amount, and then we have this heterogeneous mix of monomers, oligomers, fibrils, everything. Yeah.
Okay, there's one question. Let me ask one more question. Based on your solid state NMR data, you only measure the chemical shift, and then you talk about the fiber structure, but the chemical shift is only local Fusey angle indicator. So the structure need not be the same. So
absolutely,
you did say something. Could you elaborate on that?
Yeah, well, definitely. Yeah, I mean, absolutely. I mean, the thing with the solid state NMR, and as we just learned from Rob Tycho's work, right? I mean, like the entire community has always assumed that there is this hairpin structure for A beta 40, right? And then all of a sudden the cryo-EM showed that there's also this extended model, right? And of course one has to ask whether or not this is inter or intramolecular and all those things, right? I mean, so I think the solid state NMR is really good to prove or disprove a model that you can associate to a certain hypothesis, yeah. But a de novo structure determination is a lot of effort, and we haven't done that. I mean, we have done a little bit on that on this phenylalanine lysine mutant, because that one gave the largest perturbation of the beta strand structure, but we are far away from really understanding the high resolution structure of all our mutants. And I totally agree. I mean, they could, in principle, some of them may be hairpin, some of them may be extended or so. So this is something that at this point of investigation, we just cannot do. I mean, the cryo-EM is getting much faster, and maybe this would be a way to address this if this was really interesting to the community. Yeah.
So there's a question from Christian Grisinger in the Q&A folder. You can take a look at it. I'll read it for you. Hey, Daniel, maybe I missed it, but you said that F19 was inward in the hydrophobic core of the fibril. At the same time, it is essential for lipid binding. How can that be understood, since I would expect that the F19 would be outside to make contact with the lipids?
Yeah, no, absolutely right, Christian. The thing is, the lipid binding is something that happens on the oligomeric level, and what we've looked at was the mature fibril, and that's why these structures would be quite different. We also think that in the models, at least, that in the oligomeric structure we have intramolecular hydrogen bonds, which then sort of like expose the phenylalanine much differently than in the mature fibrils, where you have this zipper motif where the hydrophobic residues move inward. So I think this is a.
Sorry, go ahead.
Go ahead. Yeah.
Did you try changing the lipid composition to see if the fibers bind better with the liposomes?
No, I think this is so Dipta has to correct me, but I think this was always the PPC mixture, the phosphatidylcholine, phosphatidylglycerol, and cholesterol 1:1:1. So far, this is all we have done.
Okay, so Sidita raised his hand. Let me, Sam, can you let him in so that I will read the next question from Flip Neudecker. Hey, Daniel, maybe I missed this. How do you know it is the F19L34 interaction that is important for toxicity rather than a potential interaction of F19 with the membrane?
Yeah, that's a good point. I mean, we basically the way we looked at this was always coming from this hydrophobic contact and modifying the contact, and then at some point we realized that the mutations that we introduced at position 34 are not as dramatic as the mutations at phenylalanine 19. So it's not so much the contact really, but it's just the F19 side, which I think I put on one of the last slides or so. And yes, this interaction, as Christian Klösinger already asked, the relevance for the F19 for membrane binding is of course there, and it's of course much different from the structure of the oligomers and the structure of the fibrils, that this definitely plays a role. So it's not exactly the if I said this, then it's not correct. So it's not the contact or the interaction, rather I think it's really the F19 that plays a decisive role here.
So Sudipta, you should be able to speak. Can you speak up?
I will try it. Can you hear me?
Yeah, we can hear you.
Hi, Sudipta.
Okay, great. Hi, Daniel. Great talk. Thank you.
Thanks.
I was just wondering about all these things which are non-toxic or less toxic, yet the fibrils come very similar. So that would suggest that the oligomers are very different. Shouldn't that be reflected in the kinetics of aggregation if the oligomers are on pathway?
Yeah, absolutely. I mean, we didn't see a systematic variation though. I mean, for instance, the proline mutation was about 10 times slower than whatever the glycine mutation or something like that. However, in the first group of experiments with regard to toxicity, they all turned out to be non-toxic. So yeah, of course,
which
that could
might imply that it is a off pathway oligomers, which may be the toxic one.
Could very well be. Yeah, could very well be.
But your fiber morphology based on your TEM and you are talking about based on chemical shift, could be there could be polymorphism, even though they look similar in chemical shift. And a TEM.
And we see that also. I mean, sometimes we see two sets of chemical shifts or something like that. But again, it's not very consistent. It's not very systematic, right? I mean, we can only say we made our fibrils always under the same conditions. And then in some cases, we had a polymorphism at whatever valine 18. And in some cases, we had it at phenylalanine 20. But it was never really a very systematic relationship.
Well, it looks like there are no other questions. Daniel, just stick around until the end.
Absolutely.
We can discuss more. Let's move on to the next speaker, Dr. Gursky. Olga, can you share your screen? Daniel, you need to stop sharing. Yeah, great.
Yes.
Okay, our next speaker is Dr. Olga Gursky from Boston University. She received her PhD in physics and postdoc training at Brandeis University, Boston. She then moved to the Department of Biophysics at Boston University to join as a faculty. Since 2013, she has been a professor of physiology and biophysics at Boston University School of Medicine. Her research focuses on biophysical studies on lipoproteins and protein misfolding and amyloids. So with that brief introduction, Olga, get started. Thanks.
Thank you. Yeah, thank you so much, Rams. Can you hear me?
Yes.
Yes, excellent. Yeah, thank you, Rams and Magda and the rest of your team for organizing this great ZOOMinar for us, which helped us survive the pandemics. So the work I'm going to present is more like a hypothesis type study. It was performed by Emily Lukovic with the help of Shabini's GRM and the senior scientist. Emily is a PhD student. As soon as she joined the lab got closed for many months for the pandemic. So Shabini and I gave Emily a project that would keep her busy during the pandemics, and that is that she could perform remotely. And that's what I'm going to present today. So many of you know that amyloid means starch-like in Greek. The term was coined by Virchow, who noticed that amyloid-laden tissue stained with a starch stain. And he was probably looking at the inflammation-linked amyloid A amyloidosis, which back in the 19th century was predominant form of systemic amyloid disease among humans. And if so, then now we have the high-resolution X-ray structure of serum amyloid A, the protein precursor, as well as the atomic structure of amyloid fibrils. And of course, now we know that amyloids are not made of starch. They're made of proteins. By the latest counters, up to 40 major proteins and several additional proteins, which are always found in amyloid deposits, such as APE. We'll quickly touch upon it later. Amyloid deposits ex vivo always contain lipids, as well as GAGs, which stain like starch, and they may contain additional components, nucleic acids, metal ions. We'll talk about them briefly. And the question we are asking is whether these additional common constituents of amyloid deposits are innocent bystanders or active players in amyloid formation. And the answer, of course, is not only of fundamental importance, but also it is important for therapeutic targeting of amyloid misfolding pathways. So today our focus is on GAGs. Here is a tetrasaccharide unit of heparin that is often used as a highly sulfated mimetics of heparin sulfide. GAGs, of course, are key components of the extracellular matrix, but relatively little is known about the structure-function relationship in these long chains of sugars, in part because they are highly flexible and highly heterogeneous. So they are highly sulfated polyanions. All these polyanions repel each other, conferring flexibility to the chains. And we think that this flexibility in part is responsible for binding promiscuity, which amounts to large number of very diverse, completely unrelated proteins that can bind heparin, and this binding regulates protein function in numerous physiological processes, as well as in several pathologies, including amyloid diseases. So GAGs, in terms of amyloid, are, of course, ubiquitous constituents of amyloid deposits in vivo. And GAGs, such as heparin sulfate, accelerate amyloid formation in vitro, as well as in vivo. Let us start with a couple of examples of in vivo effects of heparin sulfate. Perhaps the best known is inflammation-linked amyloid A amyloidosis. And here, thanks to pioneering studies by Bob Kisilevsky, it was shown that heparin sulfate accelerates amyloid formation in a mouse model. Conversely, overexpression of heparinase, which chops GAGs into short fragments, protects the mouse from amyloid formation. And as a result of these and many other related studies, heparin sulfate interactions with SA were proposed as a target in amyloidosis. And unfortunately, the small molecule disulfonated drug, which was used in clinical trials as a molecular mimic of heparin, intended to block protein heparin interactions, failed to show efficiency in phase 3 clinical trials. And Bob thought that perhaps this molecule was too short to effectively compete with long heparin chains. And we'll get back to this idea a little later. Now, in Alzheimer's disease as well, neuronal heparan sulfate has been shown to promote amyloid pathology by modulating clearance and aggregation of A beta in a mouse model. Conversely, overexpression of heparinase in a mouse or deficiency of heparan sulfate in humans reduces amyloid deposits. And again, interactions of A beta with heparan sulfate have been proposed as one of the many therapeutic targets in Alzheimer's. And one final example in prion diseases, GAG interactions was shown to impact the structural conversion of prion to the prion secrety protein conformation and the transmission of secrety disease. So it is clear that in vivo, at least in several amyloidosis, both systemic and neurodegenerative, GAGs play an important active role in promoting amyloid formation. As far as in vitro is concerned, there are numerous examples to the same effect. Here is one from our own lab, apolipoprotein A1, the main component of good cholesterol, which is also known as a scaffolding protein on the lipid nanodiscs, highly alpha-helical lipophilic protein, which is involved in two types of human systemic amyloid disease, acquired, where mild oxidation promotes A1 misfolding into amyloid that contributes to atherosclerosis in important ways. And also there is a hereditary form, completely different disease causing organ damage that is due to point mutations in A1. As far as interactions with heparin are concerned, little is known about them. Natively folded A1 doesn't seem to bind heparin with high affinity, and the effects of it on amyloidosis have not been explored. So we began to address those, and here is a snippet of our data. Remember, mild oxidation promotes amyloid formation by the wild type protein. You can see it in the sigmoidal kinetics of CHC fluorescence. Upon addition of heparin, the lag phase in this sigmoidal growth is abolished, and the growth phase is accelerated. And this effect is pretty typical for a wide range of other proteins. Here is another PET protein in our lab, serum amyloid A. We have no time to talk about this really cute little protein, which we propose forms a lipid scavenger, whose highly conserved evolutionary role was to clear cell membrane debris from the sites of injury. So this protein is greatly upregulated in inflammation, and as the name suggests, it forms amyloid, AA amyloidosis, which is a major life-threatening complication in chronic inflammation. And a lot is known about interactions of this protein with heparin. It does bind heparin in a lipid-dependent manner, kills the protein of the lipid with binding, and also heparin binding accelerates amyloid formation by this protein in a mouse model, in cell culture, and in a test tube. And here is an example of a test tube where, again, addition of heparin eliminates the lag phase, accelerates the growth, and also influences the morphology of amyloid. So clearly, heparin binds to the protein and acts at very early stages of amyloid formation. The question is, how does it do it? So actually these and lots of other studies clearly show that heparin tends to promote amyloid nucleation and growth by a very wide range of entirely unrelated proteins with diverse native structures, which may or may not bind heparin in their native state. So the question is, how does it happen? So we propose that heparin acts as a template for heterogeneous nucleation of amyloid and stabilizes the structural motifs in amyloid formation that form bottlenecks. We were not the first to propose this template-based mechanism, but we just asked the next question. What are these structural motifs and the bottlenecks in amyloid formation? And here, of course, we were greatly aided by the resolution revolution in cryo-EM and major advances in solid-state NMR, leading to more than 100 atomic structures of amyloid fibrils now available. And these structures show that, unlike short peptides, amyloid proteins are comprised of parallel in registry beta sheets that are packed in these nearly planar molecules, one on the top of the other. Now, parallel in registry is, of course, important because here there ought to be identical residues separated by about 4.7, 4.8 angstrom. And if they are charged, such charged residue arrays of closely spaced residues are energetically unfavorable unless they are compensated by opposite charges, which is not always the case in amyloid structures. Usually the charges are compensated inside the amyloid core. On the outer surface, they may or may not be compensated by oppositely charged residues. So we propose that perhaps basic arrays in amyloid can be stabilized by binding to periodic polyanions with complementary charge in geometry, such as GAGs. In fact, the approximately 5 angstrom spacing between the nearest sulfates in heparin sulfate is comparable to the spacing between identical residues in amyloid cross beta sheets. And of course, both interacting partners are somewhat flexible, so they can adjust this spacing to match charge and geometry. So to test this idea, we used atomic structures of amyloid. And here is an abbreviated table containing listing ex vivo amyloid fibril structures from various diseases, and highlighted are the cryo-EM structures that show extra densities not accounted for by the protein model. So here are neurodegenerative diseases. The same is true for systemic amyloid diseases. This is just a small subset of all available structures, but essentially lots of them contain mysterious extra densities. And we decided to test whether or not some of these densities might represent bound GAGs, at least in principle. So to approach this, we used flexible docking of heparin to amyloid structures by using the class Pro server maintained incidentally in our school. It's a nice little algorithm that is well suited for long-range charge-charge interactions, but as any flexible docking, it has limitations. It helps identify potential binding sites and coordinating groups, but to get the exact geometry and binding affinity, you need MD simulations. So Emily used ClassPro to dock heparin tetrasaccharide to various amyloid fibril structures, and let me show some examples. So here is our favorite protein, human serum amyloid A, the cryo-EM structure solved in Fundrich's lab. If top view, so there are 2 protofilaments, and each one contains 2 uncompensated charges on the surface, basic charges, one near K46, another one near this histidine-arginine pair. And notably, there is additional extra density decorating amyloid fiber near this lysine, and a smaller density near the histidine-arginine pair. And docking of heparin clearly depicted these 2 surface sites as potential binding sites for heparin. This is actually very nice. We have done a similar exercise using a somewhat different structure of mouse SA fibril, also determined in Fundrich's lab. And that also showed extra density and a strong site for heparin binding near this histidine-arginine pair. And that's very good because this finding is strongly supported by previous experimental studies showing that this histidine-arginine indeed forms a heparin binding motif, and that protonation of this histidine actually modulates heparin-induced amyloid fiber formation by SA, and it really pops up as a major heparin binding site in mouse amyloid. So in our ongoing work with our collaborators from Straub Lab, we use molecular dynamic simulations, and the results are encouraging because for human amyloid, for instance, MD clearly shows K46 as the major, very high affinity site for heparin, and the other site, which has weaker density, as a minor low affinity site. Now let's take a look at another example, and that is creatine protein, prime protein. So here again, there are lots of uncompensated basic charges on the surface of this protein, and some of them corresponded to extra densities, and some of these extra densities have been assigned to bound glycans, and others were of unclear nature. And our docking clearly showed that the major extra density corresponds to a potential binding site of heparin, and other densities also correspond to uncompensated basic charges. And this, of course, is important because you know that in prime diseases, heparin sulfate interactions are important players in the disease progression. Now, so far, these examples show that potential bound heparin decorates the fibril surface from the outside. But in other fibrils, the polyanions such as GAG can be bound in the core. And perhaps the most striking example is human alpha-synuclein. Here you have a beautiful cryo-EM structure with two protofilaments and a large channel, aqueous channel between them with a strong extra density. The channel is lined with several basic residues and histidine, and it is clear that the bound moiety inside the channel must be a polyanion. Without it, this whole assembly would have exploded. It would be extremely unstable. So, needless to say, our docking exercise showed that heparin might potentially fit all the very, very tightly into this channel. And whether it is heparin or some other polyanionic cofactor, it is clear that this cofactor must be incorporated during the amyloid growth. It is integral to the structure of the amyloid and is crucial for its nucleation and stability. Here are other examples of tau fibrils, which, as all of you know, is highly polymorphic. It is a basic protein that binds nucleic acids. Polyanions are thought to be obligatory cofactors of tau amyloid deposition. And again, here we are looking at extra density. It's in some of the tau polymorphs, some located inside the protofilament, some between protofilaments, and some on the outside. And docking of heparin suggests that at least some of these extra densities could potentially be bound heparin, which is not to say that it is bound heparin. It could very well be something else. And if it is a polyanion such as heparin, that actually agrees nicely with biophysical studies from Sonjihan's lab, showing that fibrils dissolve when these anionic cofactors such as heparin or nucleic acids are removed. And it is clear that for the interfilament, for instance, location, that should be the case. But there is an alternative mechanism of charge stabilization, which are post-translational modifications at some of these sites, and we'll talk about it a little later. Here, perhaps, is the proof of principle. So here is a structure of the heparin-induced fibrils, which were grown in vitro from recombinant tau, which has no post-translational modifications. And they do show extra densities, most, although not all of them, near the uncompensated basic charges of the fibril surface. And our docking exercise depicts actually all of them as potential binding sites for heparin, this one being the major binding site. So we see this as a proof of concept that, in fact, some of these densities may potentially represent bound heparin or related molecules. So let us take a side view at several other fibrils with heparin docked to them. We conclude from the analysis of these and many others that predicted heparin binding sites often match extra densities seen in cryo-EM maps, that heparin molecule at these sites usually runs along the fibril axis. It is coordinated by basic residues, at least one arginine or lysine, sometimes more, like 3 basic residues here in alpha-synuclein. And importantly, the interactions between protein and heparin are mediated by coulombic attraction of heparin oxygens to the protein side chain nitrogens. We see essentially no van der Waals interactions, neither here and not much in molecular dynamic simulations. If so, then such charge-charge interactions are not expected to be terribly specific between polyanionic arrays. And if so, then maybe other polyanions can bind amyloid through similar mechanisms. Here, we took as an example an oligothiophene and an amyloid imaging dye, which is a polyanion with 4 acidic groups that was actually proposed as a potential anti-amyloid compound for A beta. By binding of this compound to the peptide, it converts apparently amyloid structure into a less toxic, non-toxic fibrils. And very similar compounds, so that the question we asked, do these compounds bind through the same mode as suggested in our heparin docking studies? And yes, in fact, there is experimental evidence from studies in Abutis lab that used one of these, actually a series of such compounds in solid state NMR and molecular dynamic studies. And they did show that the regularly spaced carboxyls in the drug form this anionic array that interacts with complementary regularly spaced cationic amyloid residues of primes. So it is a very similar binding mode to what we proposed. And they showed that a minimum of 4 acidic residues are involved for efficient binding. And that brings us back to Kayakta, which was perhaps too short with its only 2 sulfates in order to effectively bind to the site of amyloid fibrils. Now, so far, we have been talking about some unifying themes in binding of polyanions to various kinds of amyloids. But of course, there are specific effects. And where do those come from? We think that, of course, there are local differences in the specific structure on the surface of amyloid fibrils. Besides, heparin may bind not only along the fibril side, but also at the top or at the bottom surface of the fibril, which of course are unique to different amyloids. And such binding might interfere with amyloid nucleation and growth. And here is an example for serum amyloid A of a potential heparin or other polyanion bindings. Now, GAGs can also interfere with metal ion-induced amyloid formation, which is important for various proteins and peptides. Of course, they can bind to natively folded proteins. Remember this heparin sulfate proteome, lots of them. And such a binding may induce conformational changes in the proteins. It may shift the populations of native and amyloid conformations. And in general, this kind of binding can influence amyloid formation in the protein and GAG-specific manner. So yes, there are specific effects. Nonetheless, the dominant experimentally observed effect of heparin sulfate is to augment amyloid nucleation and growth in a wide range of diverse proteins. How about other polyanions? Linear periodic anions such as single-stranded nucleic acids or polyphosphates have been actually shown to promote amyloid formation by a wide range of, again, unrelated proteins, some of them basic, but not necessary, not all of them. Surface periodic anionic arrays, such as anionic liposomes, have also been shown in studies from our and other labs to eliminate the lag phase very often and to accelerate the growth. So to act in a manner that is reminiscent of the effect of heparin. And anionic detergent micelles have also been shown to promote amyloid formation by unrelated proteins. And finally, this is just a speculation, anionic lipid arrays in cell membranes which contain gangliosides, for instance, might perhaps act through mechanisms that involve this surface nucleation. Who knows? And we propose that these mechanisms through which anionic arrays can promote amyloid formation include protein condensation, polypeptide side chain orientation that favors parallel and registry beta sheet, and, as we discussed, stabilization of uncompensated basic residue arrays in amyloid with complementary anionic arrays. How about single anions? Can they promote amyloid formation? Yes. We just heard from Daniel that increasing salt concentration increases the rate of amyloid formation by A beta and actually many other proteins and peptides. The effect is particularly strong if the anions are covalently linked to each other, like in polyanions, or to the protein, like in post-translational modifications, because this will decrease the entropic penalty for anion immobilization upon binding to amyloid. And in fact, post-translational modifications such as phosphorylation, acetylation, oxidation, you name it, which changes the charge balance in critical region, such modifications may have a major impact of amyloid formation. And here are a couple of examples: tau ubiquitination, we briefly touched upon it previously, and alpha-synuclein phosphorylation. And here, let me show you a picture, top view of fibril, ex vivo fibril of alpha-synuclein with a large internal channel with a lot of uncompensated basic charge, which can be stabilized upon phosphorylation of an internal tyrosine. So addition of positive charge or removal of negative charge can help stabilize specific fibril polymorphs and thereby influence the disease target. And other post-translational modifications might play somewhat similar effects. So far, we only discussed basic arrays. How about acidic arrays, which are also occasionally present on the surface of amyloid fibrils? How do you stabilize those? Well, one way is by binding of, let's say, divalent metal ions. Here is an example from the A beta recent structure that shows a pair of glutamic acids in a ladder with a ladder of what appears like possible metal ion binding sites running along the fibril axis. Another hypothetical example of perhaps a polycation is APE and terminal domain. So APE is, of course, the ubiquitous non-fibrillar component of amyloid deposits in vivo. And its N-terminal domain has a large contiguous basic phase that binds heparin sulfate and LDL receptors. And we speculate that perhaps this basic phase may bind and stabilize acidic arrays in amyloid, such as this one, E22D23 in A beta. And of course, APE is a major player in A beta fibril formation and Alzheimer's disease in an isoform-specific manner. So who knows, maybe this is a part of the mechanism through which A beta promotes. A beta fibrinogenesis is promoted by APE. And finally, one more mechanism through which acidic arrays might be stabilized is perhaps carboxyl pairing, which was first proposed by my PhD advisor, Don Kasper, who observed it in several other periodic assemblies. So in carboxyl pairing, adjacent carboxyls may change their conformation so that they can share a proton between the two of them at anomalously high pH. And if so, they replace their mutual repulsion with an attraction through a shared proton. So potentially something similar might happen, although it has not been observed to my knowledge, but it might potentially happen in the ladders of acidic residues on the surface of amyloid. They might be oriented so that as to share protons between them. And if so, then, this helps explain why misfolding of lots of proteins can be induced at acidic pH, which may perhaps be related to the lysosomal origin of amyloid diseases such as AA amyloidosis. And finally, secondary nucleation both homo or heterogeneous nucleation of amyloid might perhaps be driven sometimes through these interactions of complementary arrays, one of which may template the formation of the oppositely charged array. So in the remaining minute, perhaps, let me very briefly summarize, the nice properties of these periodic arrays and their potential use, not just by nature, not just in pathology, but in function. And here are just some ideas, hypotheses that I want to leave you with. So perhaps these periodic arrays can be involved in self-recognition, binding cooperativity, catalyze the assembly of complementary arrays and other molecules, such as DNA repair, provide scaffolds for that, act as activators of pattern recognition receptors in immunity, as relays for signal transmission, and maybe play other highly cooperative functions. Maybe they have unusual conductivity properties and form amyloid nanowires by, for instance, transmitting these protons. And in fact, something similar was reported recently, a phenomenon of high electronic conductivity in an amyloid through whole hopping through a ladder of tyrosines. So there are potential uses of this assembly. And of course, fibril solubility, which distinguishes functional from pathologic amyloids. And here, a beautiful example is beta endorphin storage form. It contains, in amyloid, contains a buried glutamic acid that must be partially protonated. Once it gets deprotonated, then all these arrays of buried glutamics that are uncompensated becomes highly destabilizing, and the fibril dissolves into functional monomers. So with this, my time is out. So let me thank again Emily and Shabini, our collaborators who are carrying out MD simulations, other experimental studies that we do together that I didn't have a chance to talk to, other members of my lab, our funding, and most first and foremost, all of you for your attention. And thank you, Rams, again for inviting me.
Thank you very much, Olga, for a great talk. There are questions already. Please use the raise hand function if you'd like to ask the questions directly to the speaker, or post your questions in the Q&A or chat folders. Let me read these questions in the Q&A from, I think, Jan Fischau. Thanks for the talk. As you said, there are many polyanionic biomolecules, such as RNA, polyphosphates, etc. Yet it seems that heparin is one of the most efficient promoter of amyloid aggregation. Why do you think this is the case? Do you expect a specific chemistry involved with the sulfur groups, for instance?
Mm-hmm. Yeah, thank you. Yeah, that's a great question. I'm not sure I have a good answer for it. I believe that part of the reason why heparin is so important because it's so ubiquitous. There are tons and tons of it on the cellular surface. In terms of specific chemistry of sulfur, I'm not aware of it, but it is a possibility. We still don't know, since we don't have any high resolution structures of amyloids in complex with heparin. All we can do at this point is try and do molecular dynamics. And some unique features of heparin, although the same perhaps is true for DNA, is the geometry of the polymer. Heparin spontaneously can form helices in solution that vary in pitch, tighter or looser helices depending on the sugar conformation. So the contacts that I showed you are kind of show intimate contact between the sulfate residues and the fibril, but in reality you have a helix that docks to the fibril side. So sometimes there are contacts between heparin and the fibril, and then there is a gap, and then the next contact appears. So it is very hard for me to kind of be more specific in the absence of more detailed information of why exactly, whether the binding affinity is stronger, what exactly determines the binding affinity, because for that you need to have a really clear, detailed understanding of how the binding occurs. But as far as in vitro amyloid growth is concerned, I think that comparable effects for some proteins are observed for DNA binding, for instance, and heparin binding. Yeah.
Thank you. So I'm curious, did you have any examples, or did you ever observe heparin to inhibit amyloid aggregation, like promoting phase condensation, etc.?
Yeah, we have never observed it. I believe I researched literature carefully. One example that we noted is, I think for immunoglobulin light chain, people tried several different, GAGs, and some of them were more effective than others, and some had no effect. I would expect potentially that such inhibitory effects might occur because I showed the top view of the serum amyloid fibril with a large basic surface, which looks and actually is predicted to be a binding docking site for heparin. So if heparin binds there, then it would be expected to inhibit amyloid growth through, and yet we have never observe this effect. So it seems like the dominant effect is stabilization through interaction with the site of the fibril. But the inhibitory effects in principle cannot be excluded.
There's a question in the chat box from Daniel. He's in the panel. Would you like to ask the question directly, Daniel?
Yeah, sure, I can do that.
Go ahead.
Hi, Olga. I was wondering if you also tried other glycosaminoglycans, especially those that have a lower charge density, such as chondroitin sulfate or dermatin sulfate, or maybe even hyaluronic acid.
Yes, yeah, we have not tried it, at least. I mean, we tried it, we don't have any publishable data at this point. Remember, this is a pandemics project, so all of it was carried out, most of it, remotely. But other people have tried various GAGs, and there are some GAG-specific effects. So generally, GAGs tend to promote amyloid formation. Sulfation, the degree of sulfation is important. Also, GAGs can differ in, they have regions of high sulfation and low sulfation. How exactly do those interact with amyloid is unclear. Again, experimentally, yes, there are some GAG-specific effects. I have not seen any kind of general underlying theme that would help explain them. So I think here the devil is, these are good questions. The devil is in the detail. We need to have more specific structural understanding of how they talk to each other and what is the driving force. So far, it seems that the main, if not the only driving force, is long-range charge-charge interactions and not much of anything else.
Are there any other questions? Anybody wants to join the panel to ask questions to both the speakers? We are opening up the session for both the speakers. So Daniel, so in one of your mutant, you have this F19 to lysine promoted helical formation, maybe local helicity. Do you think that there's a significance of that confirmation in the very long lag time that you observed? Is it because of the confirmation, or is it because of the something else?
I think the lag time was not much longer in that confirmation. Okay. I mean, I think the perturbation of the backbone structure basically is just a response to the side chain, which we showed to be positively charged, to sort of like totally flip out of the hydrophobic interior of the fibril, and this caused this large local secondary structure change. Of course, the whole thing is across beta still, but locally at this very residue, you'll see some torsion angles which are more compatible to helix. But of course, it's not a helix in the sense that it's like it has an I+4 hydrogen bond and so forth. Right? It's just the local secondary structure.
I thought there's one other mutant other than the proline. You had, is it lysine and the L34? I forgot. There's one mutant showed a long lag time.
Yeah, in terms of the fibrillation time, the proline was very long, about 10 times longer, and the glutamate.
Okay. And
so the lysine was not so bad, really. I mean, this was a factor of 2, 3 or something, but not. I mean, in overall, even a factor of 10 is not that big of a difference, really, if you think about it, right? I mean, it's if you consider the tremendous change that you introduced by exchanging phenylalanine for a proline also. I mean, it's still surprising that it worked at all, right? I mean, if you introduce such a big perturbation.
So do you expect these findings to be similar for A beta 42? Because
good
pretty
question,
much
yeah.
What?
Yeah. I mean, there is some work that has been done. There are some mutations that have been introduced in A beta 42. Of course, now, I mean, if I started that project again, it would be you could be much more precise because we have the beautiful A beta 42 structures, of course, and now you can sort of like pick specific sites where you mutate. But I think there's a French group that also mutated the phenylalanine 19 in A beta 42, and they did also see quite tremendous alterations of toxicity. There's a +1 paper from, I don't know, some 6-7 years ago. So this seems to be an interesting residue. However, of course, in the A beta 42 structures, the phenylalanine 19 is not involved in the contact with the lutein 34. So this is all a little bit different now, of course, since we get more structural data, and yet all the work that I've introduced is based on this crude model of the hairpin, right? I mean, now we do get more structural data, especially with the work from the people recently, and you'll have to adjust those to the new structural data that we have.
Olga, I have a question for you. GAGs are super effective in cross-seeding, seeding amyloid aggregation. So do you think, is there any potential for using GAGs as possible way to avoid the oligomer formation, just speed up the fiber formation to avoid toxicity in any other amyloid cases?
Yeah, Rams, again, good question. Not sure I have a good answer. Importantly, GAGs tend to change amyloid morphology. And sometimes, since they greatly accelerate and the growth and essentially eliminate the nucleation by acting as templates, it seems like the amyloid that is grown in the presence of GAGs is less well structured, and at least in our hands for serum amyloid A, it appears that GAGs sometimes shift the population distribution to form spherical amyloid oligomers as opposed to fibrils, although in other proteins that they may induce fibril formation. So it is important to keep it in mind because, of course, amyloid morphology is a critical parameter. So again, I don't think I can give kind of a general answer here.
Are there any questions from the panel? Magda? Or Sam?
No, I'm fine. Thank you guys for nice talks. I actually have kind of an extension to Ram's question about the toxicity compared to the toxicity of the amyloid fibrils, which we study with GAG. Do the fibrils formed in presence with GAG are less toxic or more toxic? Because you kind of covered the oligomer formation and stuff like that, but do you see any differential toxicity?
Yeah, we haven't done these studies, and I'm not aware of any studies that would have checked it. So I don't know. But in principle, you are absolutely right, because GAGs have a clear and major impact on amyloid morphology. They would be expected to have a major impact on toxicity. And perhaps another important point to make is that some of the amyloid structures of ex vivo amyloid were different from the structures of in vitro formed amyloid in the presence of heparin. So, of course, there could potentially many other factors which are present in vivo and were absent from in vitro studies, but the presence of heparin alone does not necessarily drive the amyloid formation in the same way as it happens in vivo and may even differentially promote a different polymorph. So it's very difficult to kind of extrapolate into kind of more physiologically real what the effect of GAGs would be on pathology and cell toxicity. We just don't know enough.
And I have a follow-up question, kind of follow-up question. You've been mentioning ex vivo fibrils, and then you use their structures, the cryostructures, to locate the heparin. Are you aware if there was any like mass spectrometry done on these fibrils to verify what type of GAGs they are?
Yeah, that's an excellent point. For some of them, yes, for others, no. And also an important point is that even though heparan sulfate and sometimes other GAGs are ubiquitous components of amyloid deposits in vivo, the process of fiber extraction and purification for cryo-EM may potentially remove these GAGs. So, even though mass spec of the total deposit definitely shows GAGs there, it is unclear whether or not the GAGs are there at the endpoint. But again, even a small amount of GAGs, because they work, if our model is correct, they should work at a sub-stoichiometric ratio, which we, of course, and others observe in vitro, because it is one long chain that can bind to and stabilize a large stock of fibrils. Even a small amount might be sufficient.
Thank you.
I think you guys have answered all the questions and gave great talks. Thank you very much for sharing your exciting findings. Thanks for joining us. So we'll close the session formally then.
Yeah,
Okay, thank you very much. Have a good day.
Thank you, guys.
You too.
See you.
Yeah. Thanks. Bye-bye.
Bye.